Today I'm answering an Edexcel GCSE Biology Paper 2 higher tier from June 21. The population of native white-clawed crayfish in English rivers is decreasing due to an increasing population of American imported signal crayfish. Figure 1 shows a white-clawed crayfish. Which term is used to describe the American imported signal crayfish now that it is living in English rivers? Well, indigenous means that it belongs to the particular country originally, so that's not true. Yeah, non-indigenous species is definitely correct. Non-invasive, no, it's definitely invasive, so that's why that's wrong. Pathogenic, it doesn't say anything about whether it causes disease or not, so that's why it's B. American imported signal crayfish may eat white-clawed crayfish. Poor things. Explain one other reason why the increasing population of American imported signal crayfish may have caused the decrease in white-clawed crayfish. You could actually use option D to help you here. You could say that potentially the signal crayfish may bring pathogens, microorganisms, parasites which cause disease in the indigenous white-clawed crayfish, or you could say that there's competition between the species for particular food or habitat. Remember, because they're different species, it would be interspecific competition. I'm just going to say signal crayfish may carry microorganisms that cause disease in the indigenous population. The Environment Agency is developing methods of increasing the population of white-clawed crayfish in English rivers. Which term describes these methods? It's actually conservation. Hopefully you'll realise that the other words don't make a huge amount of sense because mutation is all to do with a random change in DNA. Predation is an animal killing and eating another organism. Mutualism is where two organisms help each other out. Part 4. White-clawed crayfish cannot survive in rivers with low concentrations of oxygen. Explain how eutrophication can cause the population of white-clawed crayfish to decrease. So with eutrophication, that's when you have excess fertilizers or sewage or something leaching into rivers. That causes plant growth due to all the excess nitrate ions. Increasing in the rivers, and then the plants die. Why? Due to competition for light. And these dead plants provide food for bacteria. When these bacteria respire, they use up all the oxygen, leaving the water anoxic, and that's why the lack of oxygen will therefore lead to the death of the crayfish. So a nice full answer there. Figure 2 shows an area of woodland with some small plants growing in the ground between trees. Name a group of organisms that break down the leaves and release mineral oils into the soil. I would pick fungi here personally. You could also said bacteria. The mineral ions are absorbed from the soil to the roots of the plants. Describe how these mineral ions are transported from the roots to the leaves of the plants. So we're assuming that they're already in the roots. So the mineral ions, remember, are transported in the xylem upwards. And what's going to assist that movement, remember, it's transpiration at the leaf will help move the mineral ions in the water of the xylem through the process of the transpiration stream. A scientist recorded the mean light intensity and the mean number of small plants per metre squared for 6 25 metre squared areas of the woodland. Explain the effect of light intensity on the number of small plants per metre squared. So when we have a higher light intensity, as we see in area A, we see a much greater number of small plants per metre squared. But it's asked us to explain that, so we really need to give a scientific reason why, because more light means more photosynthesis and therefore more glucose made. State one variable the scientist should have controlled to make sure the light intensity measurements could be compared. Lots of options here. You could say the same time of day, the same meter, the same person taking the readings, the same position in the area. I'm going to say same time of day. So obviously the light levels will vary hugely based on if you're looking in the morning or in the afternoon. The scientist selects an area near the edge of the woodland where many stinging nettles are grown. The area is partly shaded by the trees. Describe how the scientist should use a belt transect to investigate how light intensity affects the growth of stinging nettles. So first of all, point out how we carry out a belt transect. So that means placing a quadrat along a tape measure. Then you want to measure the light intensity at different distances from the wood. And then in terms of how it affects the growth of stinging nettles, count the number of stinging nettles along the transect in the quadrats. Figure 4 shows ciliated epithelial cells from the airways of a human as seen using a light microscope. Draw the cell labelled A in the box below. Label 3 parts of the cell on your diagram. There's the cilia, the hairs. Here's the nucleus. We could label the cell membrane. Cytoplasm. You could have also labelled the cilia. State the function of ciliated epithelial cells in the airways of the human breathing system. Remember, those hairs waft mucus. And technically that mucus is laden with bacteria. Figure 5 shows equipment used to investigate the rate of respiration in maggots. As the maggots respire, the drop of coloured liquid moves towards the test tube. So this drop of coloured liquid will move in this direction. That's because the maggots are going to release carbon dioxide when they respire, and that will be absorbed by the solar line. Figure 6 shows the position of the drop of coloured liquid after 10 minutes. Use information from figures 5 and 6 to calculate the mean rate of respiration in millimetres per minute. So we know we have a 10-minute time frame. And how far did it move? Well, it went from 0 to 2.5 centimetres. which is the equivalent of 25 millimeters. So our answer here is 2.5 millimeters per minute. Describe a control for this investigation. So what experimental procedure do we need to set up to check that it's actually the maggots causing that ink to move along? The best thing to do here is set up the apparatus as in the initial investigation, but use glass beads instead of maggots. Figure 7 shows the human urinary system. Urine is made by removing water, urea and salts from the blood. Use the letter X to label where the urine is made. It's made in the kidneys. Which substance is converted to urea in the liver? So this deamination occurs in the liver, and that's where the excess amino acids are broken down to produce ammonia and then urea. A patient cannot remove enough urea from the blood when making urine. Figure 8 shows the dialysis machine attached to the arm of the patient. Their blood is pumped out of their arm, passed through the dialysis machine and then put back into the patient. Describe what happens to urea as the blood passes through the dialysis machine. So this membrane and the fact that the concentration of urea in the dialysis solution will be 0 means that urea leaves the blood by diffusion and enters that dialysis fluid. Two patients need to have dialysis treatment. Patient A needs the treatment 3 times a week. Patient B needs it once a week. Give one reason why patient B needs dialysis treatment less often than patient A. The obvious thing is that patient B has kidney disease which is less advanced or less bad, or patient B may have a better diet. Describe how to test for glucose in the dialysis fluid. So learn all your reagents. This one is Benedict's. You need to heat it. In a water bath. And then what about the positive result for glucose? It turns brick red in the presence of glucose. Urease is an enzyme that breaks down urea into smaller molecules. Explain why urease will not break down starch into smaller molecules. This is all to do with enzyme specificity, because remember that enzyme has a special part on it called the active site, which is complementary to the substrate, which in this case, the urea is the substrate. Starch will have a different shape and therefore won't bind to the active site of urease, so no reaction will take place. Figure 9 shows the structure and actual size of red blood cells known as erythrocytes. Calculate the image size of the top view of this red blood cell if the cell is magnified by 400 times. Give your answer in millimeters. We're using this formula triangle. I am. So we're asked for the image size, which we can see is actual size times magnification. So that actual size, we're looking at the top view, which is this one here. Make sure that that unit is in millimeters because our final answer has to be in millimeters. So we take that 7.5 and we divide it by 1,000 to sort out the units, multiply by that magnification to get a value of 3 millimeters. Explain how the shape of red blood cell is related to its function. So red blood cell is biconcave dish-shaped. Why is that a good thing? Because it has a large surface area to volume ratio. Meaning that more oxygen can be transported. State the role of haemoglobin inside the red blood cell to bind to oxygen to form oxyhemoglobin. Some blood transfusions contain red blood cells. Red blood cells are stored in a solution containing 5% glucose, 0.9% salt. Explain why red blood cells cannot be stored in pure water. This is a hidden osmosis question. So if you have your red blood cell, you have pure water on the outside, it means that water is going to flood into the red blood cell, and eventually, because it has no cell wall, it will burst, which is called hemolysis. So let's write that out in full. Water enters the red blood cell by osmosis. Lack of cell wall causes the red blood cell to burst. Methods of contraception include use of hormones and barrier methods. Which form of contraception is the barrier method? So that is literally a physical wall to stop the sperm reaching the egg. So oral contraceptions, no, that's drug. Calendar method, no, that's just looking at your menstrual cycle and trying to work out when you may or may not be fertile. Contraceptive implant, that again is drugs. It's the female condom that's acting as a physical barrier. State why barrier method of contraception may be used in addition to a hormonal method, because not all contraceptive methods are 100% effective, or you could say that barrier methods have the advantage of preventing the transmission of STIs. The combined contraceptive pill contains the hormones estrogen and progesterone. State the endocrine gland that releases estrogen. O and O, it's the ovary. Explain how high levels of estrogen and progesterone in the combined contraceptive pill work together to prevent pregnancy. So estrogen inhibits the hormone FSH. And why is that good? Well, FSH is follicle stimulating hormone. It causes egg maturation. So without FSH, no more eggs can mature in the follicle. What about high levels of progesterone? Well, that inhibits the hormone LH. And LH, luteinizing hormone, causes ovulation. So if you're inhibiting LH, it means that you have no ovulation and therefore no eggs being released. So together, high levels of estrogen and high levels of progesterone will do two things. They'll inhibit egg maturation and they'll stop them being released, and therefore you can't get pregnant. Hormones can also be used to treat infertility. Explain how clomiphene therapy and IVF can improve female fertility. So clomiphene therapy, this introduces hormones to the body, such as LH and FSH, meaning that more eggs can be matured and released. IVF, remember, is all to do with obtaining eggs and sperm outside of the body, popping them in a petri dish and causing fertilization there, so that when that zygote is produced, it can be implanted directly into the uterus. For example, if your fallopian tubes are blocked, then you're still able to get pregnant. hospital patient was tested for diabetes. A nurse recorded his mass and height. Figure 10 shows these measurements. Calculate the BMI of this patient using the information in figure 10. So we need this equation. BMI is mass divided by height squared. So we do 124 divided by 1.8 squared to get 38.3, to 3 significant figures. Blood samples were taken from this patient every 4 hours. The glucose concentrations were recorded. Describe the trends shown in this data. So looking at this data, you can see that the blood glucose concentration increased from 0 to 8 hours from 100 to 139 milligrams per deciliter. Then after this time frame, you can see that the blood glucose concentration decreased at around 12 hours to 19 milligrams per deciliter. And then after 12 hours, the blood glucose concentration increased again. Remember, with describe questions, you're just saying what you see, no scientific explanation needed. This patient was diagnosed as being type 2 diabetic. Explain why the blood glucose concentration of this patient remained high from 4 hours to 8 hours. With people with type 2 diabetes, the pancreas still produces insulin, but the liver loses its sensitivity to that insulin, meaning that glucose is no longer converted to glycogen in the liver, so the glucose levels remain high. This patient did some exercise during these 24 hours. Explain how the data in figure 11 shows that this exercise occurred after 8 hours, but before 12 hours. So it's around here. You can see that the blood glucose concentration decreased at 12 hours. And why would that have actually happened? Because if the patient's exercising, it means that they need their muscles to contract. So they're going to have increased respiration to do that. And therefore, they're going to use up more glucose. A student investigated the decomposition of leaves from different types of tree. Decomposes in the soil convert. You need quite an in-depth knowledge here. It's actually C, urea into ammonia. The student collected and weighed some fresh leaves from a beech tree. The leaves were placed on top of soil in a tray. This was repeated for leaves from 3 other types of tree. The trays were kept in the same abiotic conditions for 40 days. The leaves were re-weighed after 40 days. State 2 abiotic factors that should be kept the same. So abiotic factors are non-living factors which might affect the experiment. So temperature for sure. You could have said water availability, pH of soil, mineral ions in the soil, oxygen concentration. Explain how this investigation could be improved to make the results more comparable. So just have a look at their method again. It's always good to say repeat the investigation so that you're able to calculate a mean. It enables you to spot any anomalous results as well and discount them. Figure 12 shows the mean rate of decomposition for this investigation. Which leaves would produce compost in the least time? You're looking for the highest value over here on the y-axis, so that's ash, because we can see very high rate of decomposition for ash leaves. The same investigation was extended to include the leaves of a silver birch tree. The starting mass of the leaves was 28.2 grams. After 40 days, the mass of the leaves was 19.7 grams. Calculate the rate of decomposition of the leaves. Give your answers to 3 sig fig and include units. So the rate of decomposition is the change in mass over time. So we do 28.2 minus 19.7. We know our time frame is 40 days, so that occurred over 40 days. So that's 0.2125 grams per day. And they want that to 3 sig fig. So that's why that's our final answer, and there's our appropriate unit. Explain how the concentration of oxygen in a compost bin would affect the rate of decomposition. You would find that increasing the oxygen concentration increases the rate of decomposition. Why is that? Because the bacteria, the decomposers, are respiring more. Exercise causes changes in the circulation of the blood. In an investigation, the change in blood flow to different parts of the body during exercise was measured. All the volunteers used in the study were healthy females of the same age. Figure 13 shows the results of this investigation. During exercise, the blood flow to the stomach changes. Calculate the percentage change in blood flow to the stomach in response to exercise. Give your answer to the nearest whole number. So percentage change. Make sure you're looking at the stomach. So it's the difference between these two numbers here. So just read it really carefully. So this top value up here is 1,200. And then during exercise, it decreases to 800. So that difference is 400. We need to divide it by the original, which was 1,200, and then times by 100 to get that as a percent. So you get 33.3 recurring, and we need that to the nearest whole number, so it's 33%. Suggest 2 other variables that should be controlled in this investigation: intensity of exercise, type of exercise, length of time that exercise was carried out over, the diet of the person. These should have all been kept the same. Explain the changes in blood flow shown in figure 13 that are caused by exercise. So we can see that during exercise we get a huge increase in blood flow to our muscles. That makes sense because it will be delivering more oxygen and glucose to those respiring cells in the muscle, releasing energy to enable the muscles to contract. At the stomach, just as you might expect, you would see it decrease. Why? To allow more blood to flow to those working muscles. So let's write a really full answer here. There's increased blood flow to the muscles to deliver more oxygen and glucose to cells so they can respire and release energy. And believe it or not, we're already on 4 marking points. We can say decreased blood flow to the stomach to allow more blood to flow to the working muscles. You could have also mentioned other organs, so you could have said that the blood flow to the brain remains the same. Why? Because the brain needs a constant amount of oxygen and glucose to function. You could have said that there was decreased blood flow to the kidney, increased blood flow to the heart muscle, anything sensible, but Just to show you my marking points, there's 1 mark, 2 marks, 3 marks, 4, 5, 6. Commercial growers can use plant hormones to artificially ripen fruit and produce seedless fruit, which is the name of the plant hormone used in fruit ripening. That's ethene gas. Remember, that's used to ripen bananas. Which plant hormone can commercial growers spray fruit with to produce seedless fruit? That's gibberellins, that very unusual word. The effect of applying weed killer containing plant hormones to fields of wheat was investigated. Field A, weed killer was applied each year for 5 years. Field B, no weed killer was applied for 5 years. Each field was 1,000 meters squared and all other conditions remained the same. So our independent variable, what we're changing is weed killer for field A, no weed killer in field B. Let's look at the results. So the data, field A, we can see we have a much higher yield when weed killer was applied. Comment on the effect of using weed killer containing plant hormones in this investigation. So we can first of all point out that fields with weed killer had a higher crop yield. Let's give an explanation as to why we would see that. Well, that's because the weed killer works by causing the weeds to overgrow. They die, meaning that there's reduced competition for resources, so more wheat can grow. You could have also answered in terms of stating figures a bit more. So you could say that the difference in crop yield varies quite a lot if you actually look at these numbers. So there might be another factor also affecting that wheat yield that isn't just based on whether there's weed killer or not. Figure 15 shows a pot plant before it fell over and the same pot plant 2 weeks after it fell over. Explain how hormones in the roots and shoots of the plants cause this directional growth. Okay, we're talking all about tropisms. Let's split our answer into 2 because it's worth 6 marks. So let's start by talking about what's happening at the shoot. So at the shoot, remember we need to talk about auxins, and they move to the side of the plant that has a lower light intensity, the shaded side. And then what you find there is that those cells elongate, meaning that the plant bends towards the light, therefore showing positive phototropism. The plant shoot bends towards the light. Now we need to look at what's going on with the roots. Now those roots grow downwards. They're not impacted by the light levels, as you can see, because the roots aren't exposed to the light, so it's a different type of tropism taking place this time. It's a geotropism. So at the roots, a positive geotropism is shown. That means that the roots grow towards gravity. And now let's talk about the mechanism that brings that about. This time the auxins concentrate on the underside of the root. And inhibit cell elongation. The root therefore grows downwards. So very in-depth answer here to do with photo and geotropism and how various parts of the plant respond to either gravity or light.